Chagas disease, a look into an innovative way to combat the Trypanosoma cruzi parasite, the causative agent of Chagas disease. Throughout this presentation, we aim to discuss the background and disease relevance of the disease, namely what is still known about it and why it is still prevalent in society. The target focus will discuss enzymes that are the main target of this experiment, while the results and expected outcome will show what was found during the study and what can we predict for the future. The timeline will show how this experiment aims to be conducted, while the estimated cost will represent the funding needed to conduct the study. In conclusion, we will summarize what these results mean for the future of this disease. We begin by discussing the background and disease relevance of the Chagas disease. From the table above, we can see that there has been a decrease in the prevalence, incidence, and mortality of Chagas disease from 1985 to 2020 throughout 22 endemic countries in Latin America. But through this decrease, the prevalence of Chagas disease remains quite high. Although originating in Latin America, the disease has spread through different countries and globally due to factors such as an increased number of mode of transport as well as urbanization. Chagas disease is the manifestation of infection of the Trypanosoma cruzi parasite, which is vectored by triatomine bugs. Triatomine bugs infect humans through fecal matter. Chagas disease is endemic to 21 countries, mainly continental Latin American countries, as seen in the previous slide. Clinical phases of the disease include an acute phase that lasts weeks to months, while a chronic phase can last years to a lifetime. Symptoms of an acute phase include fever, headache, fatigue, vomiting, diarrhea, chest or abdominal pain, while the chronic phase is clinically identified by symptoms such as cardiomyopathy and subsequent congestive heart failure, arrhythmias, as well as cardioembolism and a stroke. Some of the main propagators in the prevalence of Chagas disease remain under diagnosis and inadequate treatment, for which early detection is important to prevent long-term heart, nervous system, and digestive tract damage that may occur during the chronic phase. The reason Chagas disease remains a problem today is multifactorial, to say the least. Chagas disease affects poor regions, mainly with limited access to adequate healthcare services. The disease is also difficult to treat due to limited treatment options, as well as ineffectiveness of drugs during the chronic stage. Diagnosis of the disease is often overlooked, as briefly mentioned in the previous slide. The cycle of disease begins with the triatomine bug that bites an infected animal, during which time the Trypanosoma cruzi parasite may enter the bug. The bug then feeds on humans or animals and excretes fecal matter that contain the parasite, which may enter the host through cuts on the skin. The parasite is mainly spread from one person to another via blood transfusions or contaminated food. The infection progresses from an acute phase to a chronic phase during the disease. The target focus of disease treatment discusses what enzymes are the focus of the study and how they will be exploited to treat Chagas disease. The enzyme inhibition targets we have decided on include cruzane and important enzymes in ergosterol synthesis. Cruzane is a highly important cysteine protease enzyme that is essential for the survival and replication of the Trypanosoma cruzi parasite. Sterol 14-alpha demethylase and squalene epoxidase are important enzymes in ergosterol synthesis. These enzymes are important in membrane integrity and highly involved in various cellular processes such as cellular transport and signal transduction. Through the inhibition of cruzane functionality, we aim to prevent parasitic replication, disrupt parasitic invasion, and impair immune evasion. Ergosterol synthesis inhibition aims to disrupt membrane integrity and other related processes within the cell. The combination of these various enzyme inhibitors could be advantageous or disadvantageous. Advantages include enhanced parasitic cell death, reduced drug resistance, and a theoretical reduction of side effects due to lower possible dosage of each individual drug within the combination drug. Disadvantages include drug-to-drug interactions which may cause adverse effects, as well as cell cytotoxicity effects. Next, we look into results and expected outcome, mainly what was found during the study and what is expected to happen in practice. Cruzane functionality inhibition may be achieved through the use of a drug such as K777, which was tested and found to be toxic. Squalene epoxidase inhibition might be achieved through the use of terbinafine, a generally well-tolerated drug with common side effects such as headaches and gastrointestinal issues. Sterol 14-alpha demethylase can be inhibited through a drug, panzoconazole, an inhibitor that is expensive and with adverse side effects such as liver toxicity and cardiac arrhythmias. Next, we will look at the timeline of drug development, the estimated time this study will take. In the first 2 years, we aim to review existing research on the target enzymes and complete target-based drug discovery. In years 3 and 4, clinical phase trials 1 and 2 will begin. In years 5 and 6, clinical phase 3 trials will begin and findings will be submitted for FDA approval. In years 7 and 8, the safety and effectiveness of the treatment will be monitored, and we may apply for market approval and distribution of the new treatment. By year 9, we hope to begin distribution of the treatment and conduct post-marketing surveillance. The budget estimates the total amount of funding that will be needed for the study. The budget for the experiment is displayed below, with the estimated costs based on the methodology to be used throughout the experiment. The total cost of drug development is estimated to be 227 million rand. I will now conclude this presentation by discussing the consensus of the results after they were obtained for the study. In summary, the synergy of the cruzane, squalene epoxidase, and sterol 14-alpha demethylase inhibitors could prove to treat patients more effectively than current available treatments. Side effects would hopefully be reduced, but should still be monitored and evaluated. Thank you.